I apologize, they had to drop something off, so I want to press pause because I'm still limited with time on here. So I restarted it, sorry for part 2. Where we left off was the idea that there's a hierarchy of these terms. So a chromosome is made up of genes. Genes are small segments, and genes are made, these genes are what control the production of those proteins. So what we learned in the whole last chapter was the idea that the nucleotide sequence, which is on DNA, we were looking at technically genes, because each one of those genes that we transcribed and translated would have made a protein. Our analogies were very small, the simulations were very small, but these things could actually be very, very large. It would have taken us days to simulate a true protein synthesis. And then we get to a chromosome. So there are 2 variations of a chromosome. Sometimes the chromosome can be very tightly packed. It's an organizational thing. When it's tightly packed, it is called a chromosome. But when it's not tightly packed, there are times during the cell cycle when it doesn't need to be that organized. During that time, it is called chromatin. Imagine like a ball of yarn that is all wrapped around like very nicely when you first get it. That's like a chromosome. It does have a form to it. If you unravel it and it's all chaotic and everywhere, that's more chromatin. It's just uncoiled, sometimes they say. So chromatin is uncoiled DNA. So how about we define that? That is a word on here, it's number 5. You can put things like chromatin is uncoiled DNA. It's in that stage when it's not actively dividing. A cell's job is not just to divide. Sometimes your cells actually do what they're built to do. So when you are working out and your muscle cells are producing ATP, they may not be dividing. Your brain cells, they actually stop dividing when you reach a certain age. For some of you, they're still hopefully dividing, but others of you that are getting into mature ages of late 16 or maybe early 16 years old, those cells stop dividing. And so their job is just to do what they do, send impulses and send messages between your brain to your body. The DNA within the nucleus at that point is chromatin. When it condenses and coils up, it's a chromosome. Now, how does it condense and coil up? I'm going to show you an image in class, but the next word that I want you to define is the word that's on there called a histone. A histone is a protein. So it is a protein made up of repeating units of amino acids, that building block. What they do is they help the DNA coil around. It's like, maybe if you think of a paper towel, like the roll that it rolls around is like the histone, it helps it. The paper towels don't roll in themselves. There is some organization needed. A histone is the protein that helps DNA become coiled up to turn into a chromosome. So you can fill that in the words that make sense to you. Histones are proteins that help DNA or help chromatin coil up into chromosomes. Okay, we just finished the whole idea of how DNA replicates. So remember that whole thing on the process of DNA replication. I talked about why it has to happen. DNA has to replicate because you go from 1 cell to 2 cells. We're going to be talking about that process in this chapter. We assumed we go from here to here. Now this chapter is talking about what is really that process of going through DNA replication of the actual nucleus. So for this to happen, there are some times you will see chromosomes drawn as this. Very simple. And then sometimes they will even show little lines on it where each one of these lines represents a gene. So this might be a chromosome with 4 genes. You don't have any chromosomes on your body that have only 4 genes. For this diagram though, there's 4. When you go through replication, you are replicating your DNA, which remember, that's what your chromosomes are made up of. So really you are replicating your chromosomes. So there are times when now you have 2 chromosomes that are identical to one another because you replicate. You don't replicate something and it's not identical. So these 2 chromosomes would then be identical. Oh. There I go, I've got to turn it back on. I got to pause it again. I can't deal with all this technology. Alright, I'm sorry. Take 2 is over. I'll start take 3.